morning, students. Today we learn the fourth chapter in zoology, molecular basis of inheritance. Molecular basis of inheritance involves the study of genes, genetic variation and heredity. DNA and RNA forms the basis of molecular basis of inheritance. It explains how an offspring looks similar to their parents. The important terms we study here are DNA, RNA, nucleoside and nucleotide, double helix model of DNA, Chargaff's rule, nucleoid, nucleosome, packaging of DNA, euchromatin and heterochromatin. Let us start with the topic molecular basis of inheritance. DNA and RNA are the two types of nucleic acids found in living systems. DNA and RNA are the major components in molecular basis of inheritance. What are the function of DNA and RNA? DNA acts as the genetic material in most of the living organisms. It transmits the heredity from the parents to the offsprings. But RNA, it is the genetic material in some virus. The main function of RNA is it acts as messenger. Here we can see the structure of DNA. DNA is composed of two polynucleotide chains. What are the major components in a nucleotide? The main components in a nucleotide are a nitrogenous base. There are two types of nitrogenous base present in DNA, purines and pyrimidines. Adenine and guanine are the two purines present in DNA. Thymine and cytosine are two pyrimidines present in DNA. The other two components in DNA are a pentose sugar and a phosphate group. The pentose sugar present in DNA is deoxyribose sugar. The major components in a DNA are a nitrogenous base, a pentose sugar and a phosphate group. Here we can see the difference between nucleoside and nucleotide. Nitrogen base plus pentose sugar form nucleoside. The parts of a nucleoside is nitrogen base and pentose sugar. For example, adenine plus deoxyribose sugar, adenosine formed. Cytosine plus deoxyribose sugar, cytidine. Thymine plus sugar, thymidine. Guanine plus sugar, guanosine. Uracil plus sugar, uridine. When nucleoside attached to a phosphate group, nucleotide formed. The main components in nucleotide are nitrogen base, pentose sugar and phosphate group. Example, adenosine plus phosphate group adenyllic acid, guanosine plus phosphate guanylic acid, thymidine plus phosphate thymidyllic acid, cytidine plus phosphate cytidylic acid, uridine plus phosphate uridylic acid. Here we can see the structure of nucleoside and nucleotide. Nucleoside include nitrogen base and pentose sugar. Nucleotide includes nitrogen base, pentose sugar and phosphate group. Here we study the double helix model of DNA. In 1953, Watson and Francis Crick proposed double helix model of DNA. According to this model, DNA formed of two polynucleotide strands. These two strands wound each other like a spiral staircase. The backbone of the helix formed of sugar and phosphate group. Nitrogen bases form the steps of the helix. Here we can see DNA, structure of DNA in detail. DNA consists of two polynucleotide strands. These two strands are anti-parallel, that they are in opposite direction. The polarity of one strand is 5', 3' direction. Other strand 3', 5' polarity. The backbone of this helix composed of phosphate group and sugar. Nitrogen bases project inside. Here we can see complementary nitrogenous bases, that is adenine pair with thymine with the help of two hydrogen bonds, cytosine pair with guanine with the help of three hydrogen bonds. There are 10 base pairs in each helix. The pitch of one helix is 3.4 nanometer. The main points are double helix model of DNA proposed by James Watson and Francis Crick in 1953. DNA is made up of two polynucleotide strands wound around each other like a spiral staircase. The backbone is made up of sugar and phosphate. The nitrogen bases project inside. The two strands have anti-parallel polarity. One chain has 3', 5' polarity. Other has 5', 3' direction. Adenine pair with thymine with two hydrogen bonds. Guanine pair with cytosine with three hydrogen bonds. There are 10 base pairs in each turn. Pitch of the helix is 3.4 nanometer. What is meant by Chargaff's rule? The amount of adenine is always equal to the amount of thymine. At the same case, the amount of guanine is always equal to the amount of cytosine. In short, the amount of purines always equals to the amount of pyrimidines in a DNA molecule. The ratio of adenine to thymine and that of guanine to cytosine is always equals, that is equals to 1. Chargaff's rule states that the amount of purines always equals to the amount of pyrimidines in a DNA molecule. Difference between DNA and RNA. DNA is double-stranded, RNA is single-stranded. The sugar present in DNA is deoxyribose sugar. The sugar present in RNA is ribose sugar. In DNA, the nitrogen bases are adenine, thymine, guanine and cytosine. In RNA, adenine, uracil, guanine and cytosine present. In DNA, thymine present, but in RNA, instead of thymine, uracil present. Single type of DNA present, but RNA different types. They are tRNA, transfer RNA, mRNA, messenger RNA, and rRNA, ribosomal RNA. The function of DNA is transmission of hereditary characters. The function of RNA is protein synthesis. Here we study the packaging of DNA helix in prokaryotes. In prokaryotes like E. coli, DNA is not scattered throughout the cell. DNA is negatively charged, so it is held with some positively charged proteins to form nucleoids. Here we can see the structure of nucleoids in E. coli. Nucleoid composed of negatively charged DNA and positively charged proteins. Here we see the packaging of DNA helix in eukaryotes. In eukaryotes, there is a set of positively charged basic proteins called histones. Histones are positively charged basic proteins present in eukaryotes. They are rich in positively charged basic amino acid residues, lysines and arginines. Histones organized to form a unit of 8 molecules called histone octamer. What is histone octamer? Histones seen in a cluster of 8 molecules. It is known as histone octamer. Here we can see the structure histone octamer. Negatively charged DNA is wrapped around histone octamer to form nucleosome. What is nucleosome? Negatively charged DNA wrapped around histone octamer to form nucleosome in eukaryotes. Here we can see histone octamer. A typical nucleosome contains 200 base pair. We calculate the total number of nucleosomes in a mammalian cell equals number of base pairs in DNA by number of base pairs in a nucleosome. That is, number of base pairs in DNA equals 6.6 into 10 raised to 9 base pairs. Number of base pairs in a nucleosome equals 200. Then the total number of nucleosomes in mammalian cell equals 6.6 into 10 raised to 9 base pair by 200 equals 3.3 into 10 raised to 7 base pairs. Here we can see the packaging of DNA. Nucleosome constitutes the repeating unit to form chromatin. Chromatin is thread-like stained bodies. Here we can see the structure of chromatin. Nucleosomes in chromatin looks like beads on string. Nucleosomes in chromatin, we can compare it to beads on string. Chromatin is packaged to form chromatin fibers. These chromatin fibers coiled and condensed at metaphase stage and form chromosomes. Chromatin converted into chromatin fibers, then they coiled and condensed and form chromosomes. This high-level packing of chromatin requires non-histone chromosomal proteins, that is NHC. Non-histone chromosomal proteins help in the packaging of chromatin and form chromosomes. Chromatin can be divided into two types, euchromatin and heterochromatin. What is euchromatin? Here we see euchromatin. Euchromatin is loosely packed chromatin, but heterochromatin is densely packed chromatin. What are other features? Euchromatin loosely packed, transcriptionally active, and they stains light. What are the features of heterochromatin? They are densely packed. Inactive region of chromatin, it stains dark. What is the difference between euchromatin and heterochromatin? Euchromatin loosely packed, heterochromatin densely packed. Euchromatin transcriptionally active region, heterochromatin inactive region. Euchromatin stains light and heterochromatin stains dark. We discuss the first part in molecular basis of inheritance. The important terms we studied are RNA, DNA, function of DNA and RNA, then the structure of DNA, major components in DNA, difference between nucleoside and nucleotide, double helix model of DNA proposed by Watson and Crick, Chargaff's rule, packaging of DNA in prokaryotes, nucleate, packaging of DNA in eukaryotes, nucleosome, then difference between heterochromatin and euchromatin. I think that all of you understand the terms and topics clearly. Study well. Thank you.